Hello everyone, today we are going to present our project in which we developed a test kit that can measure cotinine levels in urine in order to detect second smoke exposure. Let's start with the idea of the project. The toxic substances from tobacco smoke are considered to be a major health problem and can be found easily in our daily life, but there is no specific and accessible testing method. Mostly doctors tend to ask their patients about their smoking habits and often get insufficient information. Sometimes it is too late for them to know that they have inhaled a lot of toxic substances that are dangerous and too late to treat. The main toxic substance from tobacco smoke that we are interested in studying is nicotine. When nicotine enters the body, it is converted to cotinine with 76% being excreted in the urine. So we were motivated to make a cotinine urine test kit that can be used to determine the list of toxic exposure from tobacco smoke. It used colorimetric methods and gives semi-quantitative results in order to measure the real cotinine range. Our group study discovered that derivative of cotinine called gluconic aldehyde was able to react with barbituric acid to form a polymethylene dye with a reddish orange color at a wavelength of 505 nanometer. The color intensity is directly proportional to the amount of cotinine test. All of these reactions require 4 major chemicals to become the constituents of our group reagents: potassium cyanide, calamine T, barbituric acid, and sodium acetate buffer, as shown in the figure. For the best performance of the test kit, we mixed the above components into 6 mixtures together and studied the mixing sequences as well.
The order of mixing steps was considered from the order of reaction as follows. For the experimental method, the first step was to prepare the substances. We prepared the 4 chemicals as well as artificial urine containing cotinine, with a total of 15 concentrations. Next, we measure the absorbance of each mixture with a micropipette reader. The results show that the control group, in which chemicals were not mixed, had high linearity, but mixing the constituents provides better efficiency by reducing the filling time of the substance. Based on visual comparison and linearity, it was discovered that mixture 4.2 was the most effective in terms of mixture and sequence, which makes it suitable for our testing kits. This mixture has a maximum linearity of 0.9933, which was higher than non-mixed control group. This shows that mixture 4.2 is very efficient, can reduce the filling process, can display the color range clearly, and still has the highest linearity.
Mixture 4.2 was therefore chosen as the main mixture of our project. Next step, we studied the accuracy of mixture 4.2. We analyzed 2 characteristics between run and within run. When calculating the percent CV, most of the values were slightly less than or slightly above 10%, which makes our test kit quite precise and satisfactory. Subsequently, we investigated the accuracy of the reagents. We found that the average percent recovery and percent expectation were both over 90%. This indicates that our test kits are reliable. In addition, the limit of detection was found to be 2.12 micromolar, which is close to the cutoff value of cotinine in urine. As a result, our test kit can be used to repeat current methods. Finally, we studied the stability of the reagents. It was found that after 2 months, the calibration curve showed a slight decrease in linearity, but it remained at a high level. In the end, we found that the time to complete colorimetric result was 10 minutes, which is the time we took to compare colors alongside the test tips that were provided. Based on the test tips, we divided the color level into 5 levels, according to cotinine concentration range.
Our test kit will be highly beneficial to society because it can measure cotinine accurately, has an affordable price, gives fast results, and is easy to access. It allows people in society to immediately recognize the danger of tobacco smoke and be able to avoid them before they cause other harmful diseases. Thank you.